All right. I think we overcame our technical issues. Thank you for coming today. My name is Helene Cote. I am a professor in pathology and lab medicine here at UBC and a member of the CBR. And today I will present about the research going in our lab along with two of my students. We intended to be three, but one of them is realized she's TAing at the same time, so it won't happen. We want to acknowledge that our research is taking place on the unceded ancestral territories of the Coast Salish people, and we are also meeting on their land today. We honor the Squamish, the Musqueam, and the Tsleil-Waututh peoples. The outline of today, I wanted to give you first a brief history of my path, because I started, as most of you, as a basic scientist working in blood, and then I moved on to other things, and I just thought it could be of interest to you to see how that happened and how that is possible in a way. And so I will then follow with a short overview of some of the projects that are recently or currently going on in my group. And then we'll have three short presentations. The first one with Eddie, who will be talking more about the cell and molecular aspect of things. Lulu is going to be replaced by Titania. And then Tanya herself will talk about community-based participatory research and community research. A long title, but you can see that we're going from kind of basic to more human cohort work to community-based research. So my path, I started my PhD in 1987 with Ross McGillivray. Some of you would know Ross. He was the first director of the CBR. And then I went on to do my postdoc with Earl Davey, a name that may also be familiar to you. And during that time, HIV was discovered. The first treatment came along. And then in 1996, there was a conference in town, and it was a wonderful time in HIV research. We finally had a treatment that worked, not only for people controlling their virus, but also that worked in controlling or reducing transmission of the virus during pregnancy. So these were happy days in HIV. And then I wanted to move back to Vancouver. And although I had done all my work on structure function of blood coagulation protein, there was nothing in that field. And so I changed field altogether, and I went to do HIV research. Initially, I was still doing protein structure function work. And now instead of looking at blood coagulation protein, I was looking at HIV proteins, trying to understand how resistance to the HIV drugs would develop and how this would affect the protein. However, shortly after I arrived, people who were on these new treatments started dying. And it was pretty clear that it was pretty clear they were dying of drug toxicity to the drugs they were taking to treat their HIV, but we didn't understand what the mechanism was and how or why. So I dropped everything that I was doing in protein expression and switched to this. And then a couple of years later, I was a big paper came out in New England explaining what the toxicity was about, what the drugs were, how they were, what was doing it. And then I basically never looked back. From then on, I kind of got hooked into human translational research. And given that people taking these drugs were sometimes dying, I thought, wow, what happens to pregnant women who take these drugs, to their health and the health of their babies, short-term, long-term? It's pretty, we don't give toxic drug to pregnant women normally, but in the context of HIV, we did. So I started a study called KARMA, which you will hear a little bit more after. It enrolled pregnant women, children, their babies, their children, as well as adults. And it had a control arm, so people with HIV and people without HIV. And that went on, initially looking really into pregnancy and shortly after looking into aging in general, because the mechanism of this toxicity would be expected to also affect aging process. So, and so from 2000 or so, all the way to 2020 or so, I was doing mostly cohort study work, but I continued doing more basic stuff or assay development, lab medicine type things. And then in 2020, we started what we call BCC3. So it's two cohorts, the Karma cohort and the Chiwos cohort, which I will not talk about any more than that, came together and started enrolling women living with HIV, cis and trans women, as well as control women not living with HIV. And unlike all the work that I'd done before, we were not talking about community-based research, which means community was consulted. So community being people living with HIV, women living with HIV, we were consulted right from the start about what we should do, how we should do it. They reviewed our consent form, our surveys. They gave us all kind of clues on how to do it better. They were hired to do this. They were trained in research, and that includes ethics, how to conduct surveys, how to confidentiality. And anyway, a year of training that took place during the first year of COVID. And these women, we call them our community research associates. I'm not saying it here, but since 2020, December 2020, we've enrolled 610 women despite COVID, because these included in clinic visits. And since 2022, we have two of our students. I'm sorry? Two of our students are public scholars at UBC. I don't know if you've heard of the Public Scholar Initiative. And then we went a step further in terms of our knowledge mobilization. Instead of just doing the usual conference papers and all that, we now do art-based knowledge translation, and we've done events in Vancouver as well as outside. And more recently, we are going to the sharing of individual results, which is something Tanya will talk about. So this last part of my kind of research career, if you want to say, is more community-based participatory research, which UBC would refer to in me as patient-oriented research, but we go much further than just that. Throughout this time, my group has continued to also use cell models to study all kinds of things that we cannot do in humans. So some of the current projects in the group, well, on the cell culture model side of things, we have Marie-Soleil, who's looking at the effect of these drugs on human embryonic stem cell, and we showed that it affects their differentiation, which may explain some of the issues we see in the children that were exposed to these drugs early on. Lulu is studying the effects of drugs on mitochondrial health and T cell activation. Some of these cells may impair activation, which would be a problem. And then we've had a visiting student, Michael, who's developed an assay to quantify retrotranspose on line 1. I'm not talking about that today. And then the other types of projects that are more like clinical specimen cohort studies involve Juliette, who's studying the effect of clearing HCV on your immune cells. Lulu is looking at chronic and latent viruses, so the viruses that stay with you for life, how this affects your immune aging. And this is what Tatiana will talk about today. Zesho and Edward are looking at placenta, mitochondrial DNA mutation, and whether that may explain some of the increased preterm birth that we see in HIV. And finally, the more community-based research involve what you see on the left, as well as the communication of individualized research results, which you will hear about today, and the effect of hormones on cellular aging and comorbidities. So I will now pass it on to Eddie, a very talented undergrad student from our group, who will talk about this.
Hello, I'm Eddie. I'm the undergrad student, and I'm working with Zesho, who's a PhD candidate on this project. And the project is called Placental Mitochondrial DNA Heteroplasmies in Women Living with HIV and their association with preterm birth, which is a lot of words that I will define very shortly. And this study is the more cellular based, so we're looking at cell specific instead of cohort or bigger groups. So to start, azithromycin, aka AZT, was the first HIV drug, and it's a nucleoside analog reverse transcriptase inhibitor, or an NRTI, which means that it looks like a nucleotide, but it lacks the normal 3' hydroxyl group. So shown here, it's the little yellow triangles. T shapes. It gets incorporated into the growing DNA strand during reverse transcription of the HIV RNA in the cytoplasm, but after incorporation, the HIV virus can't continue that transcription because there's no 3' hydroxyl group to bind onto. And the HIV reverse transcriptase protein doesn't have a proofreading ability, so it can't remove the nucleotide. So then HIV translation is just stopped. However, while it works really well to prevent HIV replication, these drugs can also cause mitochondrial toxicity, as Dr. Cote mentioned earlier when talking about the side effects of the HIV drugs. The mitochondrial DNA polymerase is called polymerase gamma, and it's less efficient at DNA repair than the nuclear DNA polymerases. And this means that the mitochondrial DNA replication is also affected by AZT and these HIV drugs. And with long-term use, mitochondrial mutations can build up, which lead to increased rates of mitochondrial toxicity among these people living with HIV, primarily when AZT is the only drug that's used, which is now no longer the case in treatment regimens. So I mentioned mitochondrial DNA, and it's very important for this project. This DNA, which is maternally inherited, it's about 16.5 kilobases long, and it has a mutation rate that's 100 to 1,000 times higher than nuclear DNA, meaning that it is much more error prone. Each mitochondrion has multiple copies of this DNA, and then there are multiple mitochondria in a cell, so you can have mutations that are present in some mitochondria within the cell, but not all of the mitochondria, and this is called heteroplasmy. And in this study, we're defining heteroplasmy as a mutation present in greater than or equal to 2% of the mitochondrial DNA copies that we read, which is a pretty high bar in terms of heteroplasmy studies. And this allows us to be quite confident that when we're seeing these heteroplasmies in our sequencing results, it's actually there. Heteroplasmies, like any other mutations, can cause disease. And the threshold on average for disease causing is about 60%, but it really depends on the disease causing and the mutation. So it can be lower or much higher. This flowchart covers the general reasoning behind the project. So we're also looking at preterm birth. Preterm birth is defined as birth at less than 37 weeks of gestation, and this is about 8% of births in Canada. And infants born to women living with HIV are about 3 times more likely to be preterm. And currently, we don't really know exactly why this is, but we hypothesize that it might have something to do with the antiretrovirals. We know that preterm birth can have serious effects on an infant's short and long-term health, so it's quite important that we understand this increased risk. So I previously mentioned that HIVs and the antiretrovirals can cause mitochondrial toxicity through mitochondrial DNA polymerase gamma error, and this can lead to mitochondrial DNA damage in the form of heteroplasmies, which may lead to placental mitochondrial dysfunction. This mitochondrial dysfunction in the placenta, while the child is still growing in utero, may in turn lead to a decreased supply of oxygen and nutrients and a decreased production of steroid hormones by the mother. And we know that both of these contribute to preeclampsia and intrauterine growth retention, which are 2 common causes and things that contribute to preterm delivery. We also know that active HIV viremia is associated with increased oxidative stress and chronic inflammation, and we think that these might also contribute to preterm delivery. So our overarching hypothesis is that placental mitochondrial DNA damage may be associated with preterm birth through these pathways. We're analyzing this heteroplasmy with respect to preterm birth using 140 matched samples, 70 preterm and 70 term, from 2 different studies. Currently, we've sequenced 48 eligible participants, and these participants come from these 2 groups. Karma-Preg, which is one of the first Karma cohorts that Dr. Cote mentioned earlier. This is the one that ran from 2008 to 2021, and there is no community visit, there's no community research associated. So it's the unfancy study group. And then EPIC, which is epigenetics of the placenta and complications of pregnancy. And this is run by a different researcher who has very generously lent us the samples, because this is a lot of preterm controls that we don't have in our group because of the preterm birth rate. That's just quite low among the general population. We're sequencing placental mitochondrial DNA. The placenta is this organ highlighted with that arrow, and it is a temporary organ that grows inside the uterus, but outside of the fetus, and it's actually made up of fetal DNA, and it supplies blood and nutrients and other things to the fetus through the umbilical cord. And it's important to note that it is fetal DNA, it's not maternal DNA. So what we're sequencing is essentially the children. Our methods, there's 3 large steps to our methods. The first is long-range PCR, which is amplifying the mitochondrial DNA. We do it in 2 pretty big fragments. They're about 9 kilobase pairs each. and then we gel extract these fragments. Then we sequence these fragments using Illumina MiSeq, which provides about 1,000 times coverage, which means that each base is read about 1,000 times during the sequencing, and this allows us to detect very low frequency mutations with high confidence. And then finally, we align our reads to the mitochondrial reference genome, and we analyze the heteroplasmase, and we take everything above 2% frequency or below 98% frequency as the true heteroplasmase, and the rest we discard as noise. So currently, it's important to note that currently for this study and for all the results that I'll be showing you, this is all very preliminary. We've currently sequenced only 48 participants of our 140 total. So it's a small sample size. That's a small portion of what will be a much larger sample. So just looking at the number of heteroplasmes per individual to start, we can see a very wide spread from about no heteroplasmes all the way to 6 heteroplasmes. And it looks like that preterm infants born to women living with HIV may have a slightly higher number of heteroplasmes on average, as you can see in that far left group, but it's hard to tell with this sample size. And also because the spread is so large, it's hard to detect any true effects. Next, we looked at transition and transversion mutations. So transition mutations are a swap of purine bases or pyrimidine bases. So purine to purine, pyrimidine to pyrimidine. And these are largely caused by errors in the mitochondrial DNA polymerase. Transversion mutations are a switch from a purine to a pyrimidine or vice versa, and they're associated with damage by oxidative lesions. And these transversion mutations break and distort the DNA structure by either expanding or contracting the helix, and so they're generally less tolerable in the genome. Our preliminary data on the rate of transition transversion mutations highlights a possible difference in the rate of transversion mutations in preterm infants born to women living with HIV. So as you can see, this bar is at 25% transversion versus about 20% for the other groups. However, this is very preliminary, and our full sample size will hopefully shed some more light on what's going on. Our second preliminary analysis is looking at synonymous and non-synonymous mutations. Synonymous mutations are a change of the base, but they don't result in a change of the amino acid, whereas non-synonymous mutations result in a change of the amino acid or make an early stop codon. And this is only relevant in coding regions of the DNA. The synonymous and non-synonymous distinction doesn't apply to non-coding regions. As shown in the bar chart here, the term birth group overall may have a slightly higher ratio of synonymous mutations compared to the preterm group. So if you look at the preterm group, it's in general a lower rate of synonymous mutations versus the term group, which is in general a bit higher. Since the sample size is really small, distinctions between living with HIV and living without HIV can't really be analyzed at this moment. The numbers on the bars show the number of mutations and then the number of participants, demonstrating that it's likely that the possible difference seen here isn't driven by one participant with an outsized number of mutations, but rather the fact that these two groups may actually be different. Particularly with this data, because we have to cut down from all mutations to only coding, we can't be sure of what we're seeing due to our small sample size, but we're hopeful that our final data will corroborate what we're seeing here. So as a little summary, we see a potential higher rate of non-synonymous mitochondrial mutations among the preterm infants. That is all for me. Now I'm introducing Tatiana, who will talk about Lulu's research.
Hi, everyone. So two-part presentation for me. And the first one is moving from the cell-based research more to the cohort studies as Helene mentioned earlier. And I'll be presenting the work of another PhD candidate in the lab, Lulu Cote. And her project is titled Associations of HIV, Sex, and Other Chronic Latent Viruses with Markers of Immune Aging. So the life expectancy is steadily increasing in the general population, and the immune system's age-related decline that is associated with aging can greatly impact health. So this is true for the general population and for people living with HIV. Currently, there are 39 million people living with HIV globally. However, as people stopped dying from AIDS as the medications improved, a lot of studies and clinicians observed that people living with HIV started to experience age-related diseases earlier in life than the general population. And a lot of groups started hypothesizing whether people living with HIV are experiencing accelerated aging. I will also note that here moving forward, PLWH will stand for people living with HIV. So the current hypothesis and the framework to think about accelerated aging is as follows. So it's hypothesized that the interaction of HIV medication, which are definitely improved, but still there is some toxicity, and different environmental factors can affect immune activation, chronic inflammation, which then in turn manifests in the end organs as accelerated aging. Lulu's project in particular contributes to this framework by looking into other persistent viruses that can be more prevalent in people living with HIV and how they affect immune activation and chronic inflammation. So what are the viruses of interest? So there are 7 viruses of interest for this project. 2 of them are chronic, meaning that there is a continued presence of active virus in the body, and the other ones are latent. And all the latent viruses are from herpes virus family. So these viruses are linked to age-related diseases and markers of aging individually by previous research. However, there is limited understanding of the impact of the cumulative burden of these viruses on aging and immune function, and this is what this project tries to elucidate. So a little more about these viruses. For each of the viruses individually, the prevalence is higher among people living with HIV compared to the general population. The transmission routes are different for each virus, but include body fluid, sex, and then sharing needles and blood products. And then the common associations, like clinical presentations, are cold sores, for example, for herpes virus 1, which is how it manifests activation, genital warts for herpes simplex 2, and so on, as you can see on the slide. And importantly, the only vaccine available is for the hepatitis B virus. So as for the markers of immune function that Lulu is linking the viruses to, there are 3 that I'll be talking about today. The first one being the ratio of helper T cells to cytotoxic T cells, so CD4 to CD8 ratio. Normally, it is between 1 and 3, and lower ratio is associated with poorer health outcomes. And some data previously linked lower ratios to HIV status, and of course, it declines with age. Another marker is the ratio of proliferative to senescent CD8 cells, and this is based on the expression of the CD28 co-receptor, which is a common co-activation receptor for CD8 cells. And basically, when a cell loses the receptor, the CD8 killer cell is less likely or unable to respond to an antigen. So the lower ratio here will indicate perturbed immune function. And the last marker I'll talk about today is along the spectrum of T cell differentiation, which goes from naive to terminally differentiated cells. However, today I'm only focusing on the proportion of naive cells, and lower proportion of these cells indicates worse immune function. So the objective of the analysis that will follow is to characterize these markers and associate them to a number of chronic latent viral infections and to see if there's any association with HIV status and sex. So the samples for this analysis are coming from the CARMA cohort, which stands for Children and Women Antiretroviral Therapy and Markers of Aging cohort that Helene mentioned earlier. The cohort was active between 2008 and 2021, and during the recruitment, a lot of data was collected about the participants, including the factors listed here, but many more clinical information as well. And for the cohort was recruiting across Canada and recruited over 1,000 participants of both sexes and different ages. And for a subset of participants, the subset of 376, a previous master's student in the lab, Nancy Yang, collected serology data for all the viruses I mentioned. And today I'll be talking about linking serology to immune function metrics on a subset of those 376, a subset of 97. So the design of the study was very intentional. So the full study sample you can see on the bottom left, 376, and basically it divides participants based on sex and HIV status and different age bands. And you can appreciate, I hope, the similar proportions of participants in each of the cells so that there is a balance for sex and age. And the same is true for the subsets I'll be talking about today. However, of course, the number in each cell is lower. Another thing I would like to point out is the demographic characteristics of this study participants. And this is a table summarizing the 376 participants. So you can see that none of the p-values are bolded here, and usually it is a point of upset in research, but in this particular case, it's a point of pride, because it is very important that people linked with HIV and controls are balanced with regard to different demographic characteristics that can affect our outcomes of interest. So within the sexes and within people linked with HIV and not, there are similar proportions of people who report current smoking. There's a similar ethnic makeup and similar levels of education attainment and household income. And that, of course, was achieved through very purposeful recruitment strategies. So for this subset, when we just think about viruses again, for 376 participants, females, as you can see here, had more viruses than males. So this was Nancy's finding. And people linked with HIV had more viruses than controls. The same is true for the subset I'm talking about today. And here I'm showing the virus data for the N of 97 that I'm presenting. And the way I'm showing the data is a heat map. So the viruses are the rows, and each participant is a column. And if there's a color, there's a colored rectangle, there's a positive serology for that virus. And the participants are arranged based on their increasing age from left to right. So this is controls, and this is people living with HIV. And just by looking at it, you can see more color in the pink graph. So meaning that there is more viruses among people living with HIV. We can appreciate that just kind of visually, but also statistically. So even in the subset of 97, the strand of people linked with HIV having more non-HIV viruses is true. So as for the methods, so how did Lulu look at these markers? She thought live frozen peripheral blood mononuclear cells stained for markers of interest and assessed via flow cytometry. So on to the results. The next three graphs are describing CD4 to CD8 ratio. And if we look at the sex, females are trending towards higher ratio, but it doesn't reach significance in this analysis, likely due to the sample size. People linked with HIV have lower ratio than controls. And as there are more viruses, non-HIV viruses, the ratio, the CD4-CD8 ratio decreases. So HIV status and having 4 to 6 chronic latent non-HIV viral infections are associated with the decreased CD4 to CD8 ratio. Another thing we like to do in our analyses is just to visually look at the data, especially if it's a preliminary data, to see if there are any patterns. And this is where these bubble plots come in very handy. So to walk all of us through them, x-axis is age, y-axis is CD4 to CD8 ratio. The dotted line is this lower border of a normal CD4 to CD8 ratio. And the size of the bubble corresponds to the number of viruses. The more the viruses, the larger the bubble. And the colors mean different things. On the left, it's a sex determinant. On the right, it's the HIV status. So just by looking at the data and kind of visualizing each data point, we can see that on the left, males are more likely to have lower CD4 to CD8 ratio. Most of them are below that dotted line. And on the right, those males tend to be living with HIV. All right, on to the next metric. So CD4, CD8 proliferative to senescent ratio. As for the sex, once again, we see females trending towards a higher ratio. However, it doesn't reach statistical significance in the sample, but people linked with HIV have lower ratio. And we see the similar observation as before with regards to the number of viruses. So living with HIV, older age, and having more viruses are associated with a decreased CD8, CD28 positive to negative ratio. With regards to the proportion of naive CD8 cells, so that third metric I mentioned, we do not see any association with HIV status, but there is a sharp decline with age. And then the most complex figure today, I think, is trying to bring this everything together. So here in this other bubble plot, so this is once again just visual exploration of data. X-axis is age, y-axis is CD8, CD28 positive to negative ratio. Color now means proportion of naive CD8 cells, and size of the bubble is again the number of viruses. So if you look at people clustered kind of in the bottom right, you can see that these people tend to be older, over 40, have lower naive CD8 ratio, and more viruses. But of course, we're just looking at this right now, and the final statistical tests will be done once the sample is complete. All right, so to summarize everything I just talked about. For the CD4-CD8 ratio, we see the decrease, we see univalent association with HIV status and high number of non-HIV viruses. The same is true for the CD8 proliferative to senescent ratio. And naive cells are declining with age. And for all three metrics, the effect of sex remains to be elucidated. Last but not least, another complex but very interesting figure. So here the axes are a little bit different. The x-axis is CD4 to CD8 ratio, y-axis is CD8, CD28 positive to negative ratio, color is naive cells, and size of a bubble is non-HIV viruses. And the idea here was to bring all three immune metrics with the number of viruses. And if you look at the people clustered kind of on the top right, you can see that there's a positive relationship between CD4 to CD8 ratio, CD8, CD28 positive to negative ratio. Those people also tend to have more naive cells, and the bubbles are smaller, so they have fewer non-HIV viruses. So very exciting to see what happens once the future steps are completed. And they include increasing the analysis to 350 participants. That will give us more power. And then elucidating independent effects of sex, virus combinations, and various demographic characteristics that, of course, are very important to account for. On that note, I will remain here and talk about the last project in this kind of cell to society approach. And this is more community-based research and how we are engaging with community. So as I kind of shift after that immunology heavy topic, shift towards a little bit a different direction, I would like to start with reminding everybody about a concept of helicopter research. And this is something that people don't think about often if they do the research in the local communities, right? Because usually helicopter research is a research when Western teams go to low-income countries, go there on a helicopter, maybe, who knows, extract information, samples, data, and then leave that community without integrating any findings into the community or kind of ensuring any benefit of the community. But what I really would like everybody involved in local community research to think about is that even if we're not going anywhere and our communities are right here, who is benefiting from the research that we're doing, especially if it's the research that involves people? And it's important to think about whether there's anything else we can do in terms of strengthening this bi-directional relationship between people who participate in research and researchers. Of course, this applies more to cohort studies, but I think it's important to think about. And this is what we try to think about in BCCC3 study. It's been 4 years of thinking about it. So my work in my PhD explores some of the biomedical markers and hypotheses about accelerated aging in women, and this study only recruits women, but also I do some community-based things. But first, to briefly introduce the cohort, as I said, only recruits women, started in 2020, very impressive recruitment despite COVID. As I mentioned, over 600 women. There are 2 visits that women do. The first one is at BC Women's Hospital or during recruitment drives. And at that visit, we collect biomedical, clinical information, and a lot of samples. And then there's a community visit that can be in person or virtual. And here we collect all these social determinants of health that kind of the healthcare community is recognizing are very important to account for when you're trying to understand aging. And this research survey is administered by BCC3 Community Research Associates. And I will remind that these are women living with HIV that are part of our team. They're trained in research and they're amazing. So we are trying in conducting the research, we are really trying to be feedback driven. And when somebody brings a new idea up, either participants or our CRAs, we try to act on it. And this is what happened with me. So we heard a lot from our participants, and that was communicated to us from our research associates that are doing the visits, that, you know, the informed consent process, very lengthy, very detailed, as it should be. So as women were hearing about all these amazing things we're going to do to understand their aging process and a lot of the research data we're going to generate, a lot of women were asking, oh, are you going to call me and tell me where I am with respect to that research result? And we do give to participants their clinical lab results that are done at BC Women's, but we were not until recently communicating the research results. So after there was continued interest, we decided to act on it, and we created this series of events called BCC3 Healthy Aging Talks. So the big idea was to complete the circle of the relationship between community and researchers and bring actionable findings from the BCC3 study back to the participants during knowledge mobilization events. And we hypothesized that if you really try real hard, the individual tailored research-based feedback can be effectively shared with women living with HIV and without HIV enrolled in the study. And this will help strengthen the bidirectional relationship between research and community. So this is how we approached this project. So the first step is we just sat, we had a couple of meetings, a lot of meetings with our BCC3 community research associates, and we just talked through the research data we're generating. So what it is, what does it mean? Do we know anything about how to improve it, things like that. After we identified things that make sense to communicate, we recruited and hired a woman living with HIV who enrolled in the study to work on creating these booklets and planning the events so that we have a perspective of a study participant in what we are doing. And if you're wondering where the money came for this, I will answer that question in the end. After that was done, together with that participant, we prepared individualized booklets that I will show later. And then when we were all done, we had events that I will also show later, but not too much talking. All right, so this is the big idea of kind of what we tried to do. And I will visualize it by showing one research result that we are communicating. So there is this thing we calculate in the study called 10-year risk of cardiovascular disease. We take a bunch of lab values, a couple of survey responses, and reduce dimensions and get one number that puts a woman into a category of low, borderline, intermediate, or high risk of cardiovascular, it actually should be event, I'm sorry, so a heart attack in the next 10 years. However, this is a perfectly fine figure to put in a poster, in a talk. I will probably still put it in the manuscript. But if any of our participants look at this figure, when they stumble upon it, there is no way of them to know where they are, right? Are they in the low category? Are they in the borderline? Would be good to know where you are. So what we did through iterative process, we remade this graph in a way that makes more sense to our participants. So you can see the colors are the same. The big changes is now we show each dot as a participant. You don't even need to think about axes. You just look at the background color, and then the color of the dots indicates HIV status. And if people want their personalized result, we just put a star over their dot and they know where they are. And then we have a booklet that explains better what to do about it. And I will also note that you can see that the star, yeah, you can see that it's black, so it doesn't reveal the HIV status of a person, which is of course important as we are mailing away these booklets. A little bit about technicalities and kind of the math associated with these events. So we identified 5 results we can share. There's going to be 5 booklets. They are in progress. And for each booklet, we plan to organize 3 events. So the first event is in person for women living with HIV at Ribbon Community. So this is an AIDS service organization, previously known as AIDS Vancouver, and they provide community services and supports for women living with HIV. They're located downtown. And at these events, of course, there's presentation, there's lunch, there's honoraria for women's time, there's a booklet, there's a lot of fun. So that's in person. Then we have a virtual event for women living with HIV. And this helps us involve women outside of Lower Mainland. Like at our events, we had some women from Yukon and Island, and they were very happy to connect with other women. And then, of course, we have events for controls, because not only women living with HIV would benefit from this information. So that brings us to 15 events total that I planned, and we already had 5, so that's nice. So the next couple slides are just to ease the load of thinking and just for visual appreciation of things. So these are our event posters. We had a liver event in March in person, virtual in April, and another one in May. It's not in the poster. And then we had a heart event last Monday and a virtual tomorrow. You can see in between left and right poster, AIDS Vancouver changed their name to Ribbon Community. Okay. Couple more things I would like to mention about what makes this really possible for our team. So of course, it's not just me doing this. There are community research associates, other team members who contribute and help facilitate these events. We are partnering with Ribbon Community, and their peer navigators are attending our meetings and shaping our events in the way that makes sense for their clients. We're trying to be flexible with who wants to do what. And of course, we're also conscious of the professional growth of our community research associates. So we try to swap roles and they grow as facilitators each time. We try to use our funds meaningfully. So even for catering, we try to, for our last event, for example, we discovered this amazing catering company that's located in downtown Eastside, and they provide low barrier to entry jobs for the residents of downtown Eastside. So we're trying to see also what is the best way to use our funds and how we can support businesses that do very important work. And our planning is feedback driven. So this is just how they look, how our booklets look, and I will not go into the details, but basically for liver, for example, we start with describing the functions, and I will know that every word of this booklet, it was read by our community research associates, by our clinicians, so a lot of rounds of iterations. And then we go to the research result. So there is a sufficient background for people to understand this result. For liver, we also talked a bit about medication-induced liver damage, which is under-studied issue, about symptoms of liver disease. And my personal favorite section is the example of questions that women can just take and ask their care provider and have this conversation about liver if they think they have anything to act upon based on the personalized result they get. Kind of similar framework was followed for heart health. So we talked about anatomy, then we have a result, then we have causes, symptoms, and once again, tips about like how to interpret your lab results and what questions to ask and things like that. And these booklets were very well received, which always feels great. All right, among one of the last things I will mention is about how we structure the events. So the agenda for events is similar. We start with introduction, then we have 2 rounds of trivia, true or false questions. One is before the booklet presentation, one is after, and then we collect the proportion of correct answers. And this helps us evaluate whether there has been any learning kind of before and after the presentation. Then we have a Q&A, and since event number 3, a clinician started joining us, which brought even more value to the events to help answer questions. And then we have evaluations and gift card draw. So this is a photo from Monday, where these are our team members co-presenting, our community research associates, and our team clinician is on a Zoom call. And then, of course, I hid the faces of the attendees. So for our liver health events, we had 42 attendees across 1 in-person and 2 virtual events. And of those, 37 requested personalized booklets. So I was going and finding their star and kind of moving it around, but then we were sending it to women, and they were very excited and grateful. For our event on Monday, we had 20 in-person attendees for the heart health, and then we have a virtual one where we have another 20 who are repeat. So we also see kind of the audience growing. Yeah, so very exciting line of work. And if during my talking you were wondering about where to get the money for it, I will tell you right now. So UBC Public Scholars Initiative is one way for PhD students to fund projects like that. And they support community-based projects. And the way, because it's like an official program, this also becomes a part of your thesis. So it's not quote unquote waste of your time. They are funding up to $10,000 per year for 2 years for these community initiatives. So I got that. And then after we established the framework, we got 25,000 more from UBC Community University Engagement Support Fund to fund all these projects, because as you heard, there's catering, there are all the meetings, there are honoraria, a lot of time. So time has to be fairly compensated. And for those who are interested in starting some sort of community relationship, a lower barrier to access is Partnership Recognition and Exploration Fund that offers $1,500 to just establish a community collaboration. Communityengagement.ubc.ca also has more resources for those who are interested. On that note, I would like to acknowledge on behalf of our group all the participants who ever participate in our studies and all the multiple research teams across different sites that contribute to this work. Thank you.
And if people are interested, we're on X, we're on Instagram, and this QR code leads to our website, which describes different projects as well as associated projects. Because we have the participants and the specimen, now we can tag other projects. Most of the cost is to enroll these participants, collect their data, collect their specimen. After that, doing something with it is less expensive. Any questions for any of us? Yes, Chris.
Helene, I'm super impressed by the community outreach stuff, and you always write and read things in grants, and they hardly ever take place in reality. This is actually superb. So really well done to you and your team. It's great.
It's, Dr. Angela Kaida, our team is clinical, basic, and social science and epidemiology. And Dr. Angela Kaida had really set up the stage for community-based research and how to do it well, had developed manuals to train community people. So we really profited from that and went a step further. Like this giving back result, that's a new initiative. And the whole point of community-based research is to listen to the community, and they guide what we do. They wanted their result back. I said, okay, let's give them their result back. And now they're wanting some results, like they want their telomere length and they want their cortisol level, things like that. It's going to be interesting.
Yeah, but I actually have a scientific question too.
Okay.
Pat on the back, yeah. So the CD4 and CD8 T cells, the difference, that ratio, does that at all reflect the levels of infection originally that HIV can do on those cells as related to, say, CCR5 or CXCR4 receptors?
Well, typically, almost all the participants here are on antiretroviral therapy and have undetectable levels. So that will normalize.
Yeah, no, but I realize that. But I mean, just do they have any levels? I mean, I just want to know, what are those receptor levels for entry different on those cells?
HIV only infects CD4.
And H, yeah.
Yeah. So you expect that somebody untreated with CD4, you expect their CD4 to go down, maybe their CD8 to go up.
No, sorry, I was just asking, just a normal T, CD4 or CD8 T cell, a normal person.
It's all of them, right, for this.
A normal one, yeah, a normal person, yeah.
Yeah, we were not parsing them.
No, do we know what the CCR5 or the CXCR4 levels are?
Of those participants?
No, on any, just in general immunology. Like, is there a preference for those cells? How preferential? It's CD4, isn't it? They get more infection.
T cells always have a CD4 receptor. The proportion of CXCR5, CXCR4, and CCR5 will change. Often it will change as people get treated. And some classes of HIV drugs will only work on virus that is using one of the receptors. So if I recall, it usually starts with CCR5 and it can move to CXCR4, in which case these class of medication become no longer usable in that population. We're not collecting data on that in this cohort. Yes.
I'm tremendously impressed, actually, particularly the community studies. So you could apply that sort of, if you like, information exchange technology to a whole variety of diseases. Type 2 diabetes, once you get the people with it in a participatory group, you get momentum. You can all go to the web
and you get word of mouth. You
get lists of advice, but when you read the list of advice, then it dies on the vine. But this has an opportunity of continuity to keep it going. And I think you could apply it to a whole series of diseases, quite honestly. I'm very impressed.
Now, our next step is to take our CRA, who along this project have become amazing. Like, I even gave them a statistic class for half a day, so they can read an abstract, they understand p-values and all those things. And now, like, they're gonna go and train other women outside of Vancouver and elsewhere to get involved in the research. And basically that's their work, right? They're paid for this. and they get the income. You're totally right that it could be done with any disease. It's more challenging in HIV because the people with that disease tend to be much more vulnerable. A lot of indigenous women, a lot of immigrant women, refugees, people who use substances. So a more challenging population. So to do so well in that context has been quite rewarding. I think yeah. Yes, Jamie.
Yeah, so I wonder to what extent you can give anecdotal information on what impact this has had on individuals when they do get this information back. And if you could ever reach sort of levels of significance or whatever it would take to make a case that this should be done in this context.
Talk about that. So anecdotal evidence, couple things. So we were really nervous before our first event, and I think we had 17 in-person participants, and they were like also a little bit unsure about the format. And then this Monday when we had a second event, so we had returning women, we had women bringing friends, and there is really hard for women in particular who are newly diagnosed with HIV. It's very scary to like start reaching the services and come out with their status and like come to the service organization where everybody's gonna know if you're here, then you're living with HIV. So there were a couple situations where women brought their friends and they were like really nervous, but they really liked it. And then we always say that like, hey, like, you know, don't have to fill out evaluation, don't have to do anything if you don't want, like just be here. And after we introduced the Q&A, so we have a clinician from BC Women's join just for Q&A portion, kind of hybrid component, women really liked that they were able to talk to clinician because they said that sometimes it's hard to talk to your doctor that knows you really well, knows if you have a substance use disorder, knows that maybe if you have any challenges with adherence to medication. There's like a lot of judgment that they're, you know, it's not comfortable. So they really like being able to talk to some doctor about their health kind of openly in a judgment-free way. And we had women who, previously were like, women who attended the event were like a lot of women who previously were not engaged with our community partner. We are trying to, so we had some kind of basic evaluation for these events. And now there is this thing called Engage with Impact Toolkit, which is a toolkit developed by McMaster University for formal evaluation of patient-oriented research. And it like has different domains and stuff. So it makes so our also thought is to produce some sort of data that formally evaluates the events to drive funding for these initiatives. But yeah, anecdotally, it's going really well.
Yes.
Thanks. So I have a question about the heteroplasmies. So I guess I'll ask Eddie. I know your Ns are pretty low, but have you seen any sort of like hotspots on this mitochondrial DNA for any mutations, or do you have any areas that you're looking out for based on what's already known?
So there are a couple places that we already know are mutation hotspots. There's 3 hypervariable regions in the D loop, which is the control region of the mitochondrial DNA. So we see a lot of heteroplasmies there. And then there are also a couple places throughout the mitochondrial DNA that are what's called haplogroup locations. So those are more of an ethnic identifier actually, and like an evolutionary identifier than like a place with mutations. So there's quite a few alleles there. So we see, so yes, basically the D loop in the control region, there's the hypervariables, and then several locations throughout the whole genome that are haplogroup areas where we see heteroplasmies that are not related to disease at all because they're just like an ethnic difference.
Gotcha. Okay, thanks.
Mm-hmm.
And I think when we do the analysis, we will differentiate between hotspots and pathogenic and unknown mutations. Yeah. Yes, Ed.
You guys did a really great job. Thanks a lot. Tatiana, I'm wondering if your CD4, CD8 ratios, the preliminary results you're getting and plan on getting will be correlated to markers of inflammation.
It's not mine. the ratio is Lulu's, but I can answer the question. So she already did a couple of markers of inflammation. So maybe, next year, but basically she plans to correlate this. She has a couple kind of general pro-inflammatory and then a couple that indicate the gut permeability problems that are important for people living with HIV. So yeah, TBD.
Well, that's great.
TBD.
And for Eddie, I'm sure you guys are doing this. I already know you are, but maybe you could explain carefully how you're quality assuring your mitochondrial DNA preps and that they're not nuclear DNA.
Maybe I can answer
With
that.
yeah, you want to answer.
The step where we do two long PCR fragments is to do that. It's to eliminate any pseudogene that would be peppered throughout the genome, the nuclear genome, and so there's no pseudogene that is 9 kb long, so we
Okay.
and then we gel purify because
do.
we are anal about it. So we only sequence that. Yeah.
Thank you.
Okay, thank you, everyone.